We're now going to extend some of Mendel's principles and talk about more complicated patterns of inheritance. The first pattern of inheritance that I want to look at is incomplete dominance. And in incomplete dominance, the heterozygote has a phenotype that is an intermediate between the dominant and the recessive phenotypes. So for example, flower color inheritance in snapdragons, there are multiple genes that are involved, but one of them confers red flower color. I have here a P generation, two true breeding parents, one red and one white. They're both homozygous for their corresponding version of the trait. Now, when each produces a gamete, one with a dominant allele and one with a recessive, they produce all heterozygous offspring. And these offspring are pink. This heterozygote has a phenotype that's an intermediate, kind of a blend between the two dominant and recessive phenotypes. Now normally when I cross a heterozygote, I get a 3-to-1 ratio of red to white or dominant to recessive. However, when incomplete dominance is at play, I get a 1-to-2-to-1 phenotypic ratio, which is actually the genotypic ratio as well, of red to pink to white. And so this complicates inheritance just a little bit by altering that F2 generation phenotype. Had Mendel ran into incomplete dominance instead of complete dominance, he would not have seen his 3-to-1 ratio that he did when he looked at, for example, purple flowers versus white flowers. Another example is high cholesterol or hypercholesterolemia. In this case, there is a receptor protein, which is of course encoded by a gene because it's a protein. And the dominant allele encodes the functional receptor. The recessive allele encodes a non-functional receptor. Either it doesn't work once it's in the plasma membrane, or it's actually not present. It's not even able to be made. Normally, if you've got all the proper number of receptors, you bring in cholesterol from the blood. We want that cholesterol, of course, in the cell, and the cholesterol is removed from the blood and brought into cells and doesn't accumulate within the bloodstream and cause plaque. When you are homozygous for the inability to make it, cholesterol levels in the blood remain high, which is very dangerous and leads to heart disease, heart attack, and stroke. If you're heterozygous, you have an intermediate phenotype, which is a mild form of high cholesterol disease. You have some receptor, but not a lot, and so there is still some cholesterol remaining in the blood as compared to those that are homozygous dominant. This is another example of incomplete dominance because the mild disease is considered to be sort of a blend between normal conditions and severe high cholesterol. There are two other extensions, codominance and multiple allelism, that I can explain in one example. Codominance is when the heterozygote displays the phenotypes of both alleles. Multiple allelism refers to the fact that there are usually more than 2 alleles for a given gene within a population. Still, though, the phenotype depends on the combination of 2 alleles. However, there are more than 2 out there in the population that can produce that phenotype. An example is blood type in humans. There is a gene that encodes what we call an antigen on the outside of red blood cells. The allele IA encodes the A-type antigen, which is actually just a carbohydrate that's bound to the membrane of these cells. The allele IB encodes a different form of the carbohydrate. The allele i, or the recessive allele, is the production of no carbohydrate. Now, the presence of any combination is fine on red blood cells. But if, for example, you are A blood type, you make antibodies against any other type of carbohydrate. So, for example, against carbohydrate B. So if I'm A-type blood and you give me any blood that contains the B carbohydrate, it will clump and it will cause death. Now notice that an individual who is O has neither type of carbohydrate, and I can take O type if I am A type because there are no antigens on the outside of the red blood cells for me to clump. And so as an A individual, I can take either A or O blood. A B individual can take O or B. And then an AB individual who is heterozygous for A and B can take all types of blood because there are no antibodies made against any carbohydrates. If there were, this individual would attack their own blood cells, and so there are no antibodies produced. Now notice the genotypes for these blood types. To be blood type A, you're either homozygous for IA or you're heterozygous, IA with the little recessive allele I. Little I is completely recessive to either IA or IB. So the same condition exists for being B-type blood. You're either homozygous for B or you're heterozygous. The heterozygous condition of A and B, IA-IB, gives both types. Notice that if you're IA-IB, this isn't an example of incomplete dominance. There's not a blend. Instead, you actually see both types, both phenotypes of the two alleles. And then somebody, of course, who's homozygous recessive makes neither type. This person can only accept O-type blood. And this happens to be the most prevalent type in the population, counterintuitive to the fact that an O individual couldn't take any other type of blood, but for other reasons, O-type is the most prevalent in the population. So this is an example of codominance because of the display of both phenotypes in the IA-IB individual, and it's also an example of multiple allelism because there are 1, 2, 3 alleles in the population, and it complicates the study of the inheritance of genotypes, the inheritance of blood types. Another example of incomplete dominance is sickle cell trait. I told you guys that if you are homozygous for the sickle cell allele, you have sickled cells. These are all examples of sickled cells. And if you are homozygous for the normal allele, you have normal red blood cells, and they vary in shape. If you're heterozygous, you actually have sickle cell trait. And sickle cell trait isn't the production of blood cells that are sort of sickled. Sickle cell trait is what you're looking at here, the production of normal cells and sickled cells. So if you're heterozygous, you don't have sickle cell anemia. You also don't have normal blood cell type. You have a blend, or not a blend, a mix of the two different types. And that's why this is an example of codominance. The heterozygote displays both normal red blood cells and the sickled red blood cells. Now, sickle cell anemia is also an example of something called pleiotropy. Pleiotropy occurs when one gene produces multiple phenotypic effects, and this is the case for a lot of genes. For example, if you look back at the slides for PKU, describing PKU, having PKU, which is a recessive disorder, produces multiple problems. Primarily mental retardation early in development, but it also produces low levels of melanin. Turns out that it also affects levels of adrenaline. So this is the case where one gene is producing multiple phenotypic effects. Well, that's the case here as well. Not just the production of sickled cells, but all of the other things that happen when these sickled cells are present. Yes, you have anemia, because the cells are broken down really easily, but you also have all kinds of other effects from the sickled cell shape, pain, and fever because your immune system is reacting to the source of the pain. There is all kinds of damage to organs, failure of different organs, and damage, brain damage, spleen damage, and it's all because of the presence of these sickled cells. That's multiple phenotypic effects, just because of the presence of one particular type of allele, that which produces sickle cell. So in the case of pleiotropy, a single gene affects multiple characters. Now, it is also the case that one character can be affected by multiple genes. This is quantitative inheritance or polygenic inheritance. Take, for example, the inheritance of skin color. There are at least 3 genes involved in the inheritance of skin color. These involve proteins that are actually enzymes leading to the production of different pigment molecules. If you're homozygous dominant for all three, you have very dark skin. Homozygous recessive, you have very light skin. Now, notice that if I've got somebody true breeding for recessive and true breeding for dominant, they're going to produce offspring that are triply heterozygous. Triple heterozygous mating produces a Punnett square that's actually 64 squares large, because each one of these individuals can produce 2 to the third or 8 unique gamete types. Big A, big B, big C, big A, big B, little c, big A, little b, big C, and so on. If you write them all out, there are 8 combinations. And so 64 total combinations of offspring genotypes. Within this combination, these combinations of alleles, the more dominant alleles, the darker the skin color. One trait, skin color, affected by multiple genes, leads to this varying range of skin color. And this is the case for a lot of traits, especially those that we see, like eye color or hair color. There's not just one gene involved. The products of multiple genes can actually interact with each other to produce multiple blends, or one gene can control another, such as the case for epistasis, where one gene controls whether or not another is actually expressed. So in the case of polygenic inheritance or quantitative inheritance, multiple genes affect one character, such as skin color. This is basically the opposite of pleiotropy, where one gene caused multiple phenotypic effects or affected multiple characters. And last, remember that the environment can affect phenotype. This is certainly the case for behavior. Certainly lots of physical qualities to people. Here we have 2 identical twins that look somewhat phenotypically different because of the environment, maybe exposure to sunlight, nutrition, artificial environment like changing your hair color. While things like behavior have been shown to be controlled by genetics, certainly the environment can affect an individual's behavior. Now, a really cool example of an effect on phenotype is an example where the temperature affects the phenotype, the color of coat in different types of mammals, like for example in these Siamese cats. Temperature can actually affect the coat coloration in Siamese cats. Places where Siamese cats have darker fur color is due to the fact that the temperature is cooler in the extremities. There's an enzyme encoded by a gene that converts white pigment to brown pigment. A chemical reaction converts white pigment to dark brown pigment. Where the temperature is cooler, the enzyme is intact and can function. And so white pigment is converted to dark pigment. That's in the extremities here. Closer to the core body, the temperature is warmer, and the enzyme is actually denatured. The temperature is too high and there's too much energy, heat energy, to allow this enzyme to stay intact. It denatures, it falls apart as far as its bonding, and it can't produce brown pigment. So the reason why these white areas appear is because of a lack of pigment. Not because the pigment isn't encoded, but because the enzyme that creates the pigment isn't functional. This is the same effect in harbor seals, which are born white. They develop in their mother where the temperature is very warm, but when they develop their adult coat out in the cold, the coat is brown. The idea is that the white seal can blend in with the environment when it's a baby, when it can't take care of itself, can't protect itself. Its only protection is its mother and the fact that it can blend into the environment. As an adult, it wants brown coat, because that protects it from UV rays, and as an adult it can protect itself and so doesn't need to blend into the environment. So when it's born initially, it has a white coat to protect it, camouflage it, and then as an adult it has a brown coat in order to protect itself from UV rays. So these are just a few of the ways in which inheritance is altered by extensions of what Mendel proposed. What we're going to do next is talk about the extension of inheritance of genes that are located on sex chromosomes, because inheritance of sex-linked traits is also different, although still based on Mendel's principles.